Hello everybody, and thank you for joining our webinar. I'm Susan, application specialist for protein production and assays at IBL Life Sciences. And I'm pleased to talk about a frequently and extensive question in the field of protein production. And I thank our participants for joining our webinar dealing with this question, and I hope my explanation meets your expectations. So we want to talk about how to plan the expression of a recombinant target protein and the subsequent purification by affinity chromatography. The aim of the webinar should be that you are able to make the right decisions regarding the expression host. So if you take E. coli, mammalian cells, insect cells, or yeast. Regarding the buffer system, for example, tris buffer, MAPS buffer, HEPS, or sodium phosphate. Regarding the affinity tag, like the his tag, GST tag, flag tag, or the rubber strap tag 2 or twin strap tag. And of course, regarding the resin format, so if you take a pre-packed gravity flow column, spin columns, magnetic beads, or cartridges. To make the right decisions for the expression host, buffer system, affinity tag, and resin format, you have to find the answers for two initial questions. The first question is, what are the properties of the target protein? And the second, for which subsequent application is the protein required? Depending on the existing knowledge, the questions are more or less easy to answer, but if you take the time to answer them, the answers are a great help for your protein expression and purification. So let us begin with the first question. What are the properties of the target protein? And I will explain why this information are helpful for planning protein expression and purification. One property that can be quickly identified is the organism from which the target protein originates. That can be a soil bacterium, a specific plant, an insect, mouse, or something else. A second property that is connected with the first property is the localization within the organism, or more precisely in the cell. So if it is, for example, localized in a membrane, in a nucleus, or cytoplasm. If the localization is not known, you can use prediction tools which use the amino acid sequence to predict the localization of the protein. These prediction tools or similar tools can also be used to identify specific domains or signal sequences. That should mean if the protein contains one or more membrane domain, secretion signal, metal binding site, or localization signal. Domains and signal sequences can be an indication for a protein class, so if it is a membrane protein, a metalloprotein, or an antibody. And the two last properties I would like to mention are the size or molecular weight and the isoelectric point. So if you collect all this information about your target protein, you are well prepared for further planning steps, because all this information are helpful to select the right host, a compatible buffer system, and appropriate affinity tag, as well as a corresponding affinity chromatography resin. The knowledge about the signal sequences and organisms are necessary for the selection of the host. The localization, isoelectric point, domains, and protein class are necessary for selection of the appropriate buffer system. Signal sequences, size or molecular weight, protein class, and specific domains are necessary for the selection of the tag and the appropriate affinity chromatography resin. And in the next slides, I will focus on each circle of this diagram, and I will begin with the selection of the host. The most commonly used hosts are E. coli, yeast cells, mammalian cells, and insect cells. And yeast cells, mammalian cells, and insect cells are eukaryotic hosts, and E. coli is a prokaryotic host. E. coli cells are easy to handle, so you do not need special equipment. They grow really fast. After transformation, the protein can be harvested after just a few hours, making the host really economical. But if you want to express an eukaryotic protein, there are some challenges because E. coli cannot splice the mRNA, so you have to use a coding sequence without introns. Furthermore, it has a different codon usage compared to eukaryotic hosts that can cause an reduced yield or incomplete translation. E. coli do not have a machinery for post-translation modification, which are necessary for a functional protein. And after preparation, the sample contains endotoxins, which could be problematic depending on the subsequent use. And if your protein is produced at a high level, the formation of inclusion bodies or degradation is possible. And eukaryotic host, which shows like E. coli, a rapid growth and is really economical, is baker's yeast. Advantages of yeast are compared to E. coli that yeast has a machinery for post-translation and modifications like glycosylation, and after preparation the sample is free of endotoxins. But like E. coli, it has a different codon usage compared to higher eukaryotes like mammalian cells and insect cells. And the post-translational modification can cause a hyperglycosylation, and the protein secretion of yeast cell is very inefficient. Due to these disadvantages for proteins from higher eukaryotes, it is recommendable to work with mammalian cells or insect cells. There the proteins are correctly folded and naturally glycosylated, but mammalian cells and insect cells are not easy to handle. Special equipment is necessary, raising the costs, and contamination with animal viruses are possible, and especially for insect cells, the expression is not continuously, so you have to transform from time to time the insect cells new. To bring everything together, or to sum up, I would recommend to choose a host that is similar to the original organism of the target protein, since that guarantees a correct folded, modified, and secreted protein. So now we have finished the first cycle and go over to the next circle, the correct buffer system. At the beginning, I mentioned that it is necessary to know the isoelectric point or PE of the target protein to select the correct buffer conditions. The PE is the pH at which a molecule carries no net electrical charge, is electrically neutral, and it can affect the solubility of a molecule at a given pH. That means at the pH that corresponds to the PE, the protein denatures or precipitates out of solution. At the pH below the PE, the protein carries a net positive charge, and at the pH above the PE, the protein carries a net negative charge. So the buffer should have a pH below or above the PE of the target protein. If you do not know the isoelectric point of your target protein, you can use several online available tools to predict the PE of your target protein. If you know the isoelectric point of your target protein, you can easily select an appropriate buffer system. Each buffer system has a pH range for which is suitable. The most common used buffer system is Tris buffer due to the neutral pH range. And if a buffer system with an acidic pH is necessary, mass or sodium phosphate buffer or MOPS buffer can be used. And for basic pH, the lower buffer systems are applicable like Hebris, Trisina, but please keep in mind that the buffer system is also influenced by the subsequent application, but I will come back to this point later. After the selection of the buffer system, you should think about additives which stabilize your protein, protect it, or make it soluble. Therefore, you need the information about the localization, protein class, and specific domains because they can help to select the necessary additives. For example, metalloproteins needs metal ions, or protein with ATP binding site needs ATP to be stable. Cytoplasmic proteins containing cysteines need reducing agents to prevent unnatural disulfide bonds because on the cytoplasm, disulfide bonds are normally not formed. Some proteins have a protease recognition sites to prevent the cleavage. Metal chelators like EDTA are added to bind the metal ions which are necessary for an active protease. And if your protein is a membrane protein, you should add detergents like Triton or SDS to make your protein soluble. So now you know how you can select an appropriate buffer system and what you have to look for. And so we can focus on the last circle, the selection of the tag and the resin. To choose the optimal site for the affinity tag, information about the protein class, signal sequences, and specific domains are necessary. The picture shows only membrane proteins, but nevertheless it says more than 100 words. So some proteins have a part which is integrated into the membrane. Some proteins are partially secreted via the N or C terminals, and we should keep in mind that signal sequences are mostly cleaved off during the translocation. Some proteins have several transmembrane domains. Some proteins are complex with other proteins or have sites for cross-translational modifications. So what I want to show with this picture is that the tag should be localized on a site which is not cleaved, modified, or integrated into a membrane. The selection of the tag and the resin is also dependent on the size or molecular weight of the target protein because the affinity chromatography resin consists of agarose beads and mostly the agarose has a concentration of 6%. Due to this high concentration, the pores of the agarose beads are small and allows only the enter of small proteins. Whereas large proteins cannot enter the agarose beads and flow through. That means if large proteins should be purified, the yield is influenced by the concentration of the agarose. So to obtain a higher yield for large proteins, or if you do not want to think about the resin's properties, it's recommendable to use an affinity chromatography resin with a 4% agarose. Due to lower concentrated agarose, small proteins as well as large proteins can enter the agarose and do not flow through. So 4% agarose like streptotin XT4 flow enter a high protein yield for small and large proteins. We have focused on the first question and now we can go over to the second initial question: for which subsequent application is a protein required? The second initial question is an important question because the subsequent application defines the required amount of the target protein, the purity, the affinity of the tag, the buffer composition, and if the target protein has to be functional or active. The most frequent applications are detection in Western blot, characterization assays, interaction studies, immunization, and crystallography. For Western blot and immunization, only small amounts are necessary, but for crystallography, characterization, and interaction studies, mostly several milligrams are necessary due to the several test conditions and replicates. Nearly all applications need a high purity, especially immunization and crystallography, which needs a very high purity. A high affinity of tag is required for Western blot characterization and interaction studies because that ensures robust and reliable data. In addition, analytic applications allow only specific buffer compositions, so if the purified target protein is already present in a buffer compatible with the analytic application, the time for buffer exchange can be saved and the functionality or activity of the protein can be retained. And a functional or active target protein is necessary for structural analysis, characterization assays, and interaction studies. And you should keep in mind that for immunization and crystallography, large tags often have to be removed. Now we have learned which specifications are necessary for analytic applications and can look at the affinity tags and check which one is compatible with most analytic applications. The highest yield are obtained with the his tag, GST tag, and the strep tag 2 and twin strep tag. But the purity is only satisfactory for the GST tag, strep tag 2, and twin strep tag. The broadest affinity range is provided by the strep tag 2 and twin strep tag, and the highest affinity by the twin strep tag. That means for characterization and interaction studies based on BLE or SPR, the twin strep tag is the most suitable. The most variable buffer compositions are possible with the flag tag, strep tag 2, and twin strep tag. The GST tag do not allow denaturing agents and needs reducing conditions, and the his tag do not allow tris and abs buffer and is not compatible with reducing or chelating agents. That means proteins that are purified with the his tag or GST tag can be functional if the buffer composition suit, but the size of the GST tag can interfere with the structure and function. Furthermore, for the GST tag, only the N-terminus is recommended, and the tag is not compatible with proteins containing disulfides. His tag purified proteins often contain contaminations of co-purified proteins with histidine repeats, and purification results from mammalian and insect cells are not satisfying since the supplements in the medium or the pH of the medium reduce the resin's binding capacity. For the flag tag, if the target protein is diluted by pH shift, the protein function and structure can be influenced. In contrast, the strep tag 2 and twin strep tag are easy to use, adaptable, and the buffer composition can be tailored to the requirements of the target protein and the analytic applications. So the strep tag 2 and the twin strep tag show the highest compatibility with analytic applications like Western blot, interaction studies, characterization assays, and immunization and crystallography. The last question of the webinar is, which resin format should I choose? This question can be answered by the required yield. If only small amounts in a microgram range are required, the application of spin columns or magnetic beads is recommendable. If large amounts are required, pre-packed columns or suspension for column packing or cartridges for FPLC or HPLC devices are recommendable. To sum up, for the selection of an appropriate expression host, choose a host that is similar to the original organism and consider signal sequences. For the buffer system is given by the target protein and the subsequent application. For the affinity tag, use the most flexible tag, the strep tag 2 or twin strep tag. And for the resin format, choose a resin format depending on the required amount. At this point, I would like to thank you for watching our webinar, and if you have questions, please feel free to contact me. I'm here to help.